My name is Moses Masika. I'm a virologist at the University of Nairobi School of Medicine. I spend most of my time teaching medical students about viruses and also conducting research on infectious diseases. So today we shall talk about the process of making a vaccine as well as the challenges of delivering the vaccine after successful manufacture or development. So the process of making a vaccine is quite lengthy, and in brief it takes about 4 major steps. The first step is what we call preclinical development. Preclinical development looks at, say for example, COVID-19, the virus, the characteristics, and then identifies which parts of that virus would be used to make a vaccine. And then that is tested on small animals like mice, sometimes tested on larger animals like primates, and after that the approvals are requested using the data generated to take the vaccine to human or clinical trials. So the aim of the preclinical stage is to come up with a product and prove that it works, in eliciting some form of immunity, and also at least it has a potential to elicit immunity, and also it was likely to be safe to try in humans. The preclinical development stage can be quite lengthy because it involves understanding the basic science of the virus, how the virus looks like, what is the genome like, what are the structures on the surface, and also coming up with a product using that knowledge, and then testing it in the lab. From the lab, several products may fail, a few may show potential, and those are then taken to smaller animals like mice and guinea pigs for testing. And usually there we look at the safety of that vaccine, because if it harms the animals, then it's considered to be harmful in humans and may not proceed. So we are looking at the potential for safety in humans and the potential to induce an immune response in those animals. And after that, you test it on larger animals like primates, which are more like human beings in a way. So again, looking at, is there potential for immunity, to induce immunity or protection, and is there any safety concerns? At that point, if a few candidates are selected, and then the candidates that are selected will then get data for that, and then that is taken to authorities for approval to do human trials. There's a stage in between that involves optimizing manufacture of the vaccine. So sometimes if a product has shown to be a good candidate for taking to human trials, you need to have a manufacturer ready who is able to produce that vaccine in consistent quality and in large enough amounts for testing in humans. And some of the vaccine candidates actually fail at that step if manufacturing cannot be optimized. So it's a process that in some cases can take 10 years. In the case of COVID-19, this took a few weeks in some instances, a day for one of the companies, and others for other vaccines it took even 10 years or even longer, because it depends on how much funding is available, how many people are working on it, and the complexity of the virus. So we see some viruses, it's more difficult to develop vaccines. The good thing with COVID-19 was that the virus that causes it is similar to other viruses, 2 other viruses that had occurred before that scientists were working on. This is SARS-CoV, which occurred in 2002, 2003, as well as MERS, Middle East Respiratory Coronavirus, which occurred about 2012, 2013. And these viruses are very similar to the current one, so the technology that was used for these previous infections has been availed for COVID-19, and that informed part of the speed or the ability to produce this within such a short time. Additionally, there was a lot of resources directed towards COVID-19, so a lot of people were working on the same thing, a lot of labs, a lot of companies were focusing on this with a lot of support from governments, and so this helped in getting the vaccine out faster. So the preclinical stage can be quite lengthy, but we've seen with COVID-19, if people come together and a lot of focus is given on this, then there may be opportunities to get vaccines much sooner. So the second process can be termed as the clinical trial phase, and this has 3 phases. The first is phase 1, let's just call it phase 1 because that's what it's called, is to look at safety. And here we test a few dozen people. You could take tens, 50, 60, 80, to test them by giving them the vaccine and to see if the vaccine is safe in these persons. So you don't start with so many people. You start with a few people, maybe 5 or 10, and then as you build up, you can go up to a few dozen. And you're giving them, first of all, you explain to them what the vaccine is about, and if they consent and agree to take the vaccine, given the risks, then you administer the vaccine and follow them up. So typically, you follow them up for a year or 2 to see if the vaccine had any effect on their body, and we usually look at the liver, the kidneys, other parts of other systems in the body to see if this vaccine is actually safe. A secondary objective usually is also to see if the vaccine induces antibodies. Antibodies is a marker for protection. You want to see if the vaccine has the potential to protect against the pathogen or the germ that you're making the vaccine for. And this is a secondary objective for the first phase of clinical trials. The second phase of clinical trials occurs after you have shown in the first phase that the vaccine is safe. It does not induce any unacceptably high side effects. There may be some acceptable ones like mild fever, pain at the site of injections. Those ones are allowed, and you tell the volunteers that you're likely to have some pain, of course, because you're being injected. You may have some fever. So these are expected. But if the vaccine causes more than acceptable side effects, then it fails at phase 1. Also, before you move to phase 2, typically you require authorization from regulatory bodies in whichever country you are at. So if a vaccine candidate is successful in phase 1, meaning that it has been shown to be safe for use in humans, then the persons developing the vaccine will get approval from regulatory bodies to do phase 2. And phase 2 clinical trials are usually conducted in about a few hundred volunteers. This could be 200, 300, 400 people, and usually in maybe a few sites, maybe 1 or 2 countries or several countries, just to see if the vaccine again is safe. So you're collecting more safety data, and also if the vaccine is able to induce antibodies or protection against the virus in this case. So at this stage, you're looking at safety, but additionally, you want enough data to show that the vaccine can produce antibodies or immunity. Because if the vaccine is able to induce immunity, then it will be able to do its job. It may be safe, but it's not inducing the immunity that you require. And this is done in over another period, maybe 1 to 2 years, depending on the type of immunity that the vaccine is supposed to induce. This is a process that is done, as I said, in several sites, so that you can compare how different people also are likely to respond to the vaccine. And that is important because we know people respond to medical products differently. And as you collect the data on safety, you collect the data on ability to induce immunity, you can also collect data on what is the right dose, how should the vaccine be administered. Do you give it into the muscle? Do you give it into the skin? Do you give it through any other route? And also, what is the dose? So you optimize the dose also at this point, because you could decide to give a lower dose and see how it works, and compare with a higher dose and see how it works. Generally, if you can stay with lower doses and give the same immunity, that is safer, because you don't want to give too much of a product if you can avoid it. If phase 2 is successful, meaning that you've shown that the vaccine is safe and it induces immunity, or it is doing what it's supposed to do, then you can move to phase 3, again after obtaining regulatory approval. Phase 3 is much larger, much broader, and the key thing in phase 3 is what we call efficacy. You want to see, does this vaccine work in real life conditions? And you still additionally collect safety data, because you are now doing this in thousands of people. You've seen some of the COVID-19 phase 3 trials being conducted in 40,000 people across different countries. And that is the idea. You want to test this vaccine on diverse group populations in different parts of the world. You also want to give it to diverse ages, mostly people above 18 years, all the way to the what we call senior citizens, because different categories of people will respond differently. And you also want to give this vaccine to people who are exposed, like in this case to COVID-19, so that if you can compare and see whether those who got the vaccine versus, we usually give something we call a placebo at all stages to compare. You want to see if the people who got the vaccine are actually protected or not. And to do that, you need to have people who have been exposed, so that you see the vaccine was able to protect against that exposure leading to infection. So phase 3 trial is a large human trial of a vaccine candidate done across tens of thousands or thousands of people, mostly across different populations. And at the end of the day, you want to analyze the data and see, is this vaccine effective? Does it work? Does it prevent people from getting the infection? Additionally, because you have a higher number of people, you observe for safety. And this is the time we get to see very rare side effects, the time we may get to see rare adverse events of the vaccine, which is important because, again, that helps you to determine whether this vaccine should be licensed for human use or licensed for general use. If phase 3 is successful, then you show that data to regulatory bodies. In Kenya, we have the Pharmacy and Poisons Board. The US have Food and Drug Agency. Europe have EMA, European Medicines Authority. So all agencies, all these are regulatory bodies that will look at the data that you've produced and decide whether the vaccine is safe for general use. And if they are convinced, then they license you to commercialize that vaccine. You can sell it or whatever it is, so that people can now get it. And generally, governments are the leading consumers of vaccines, because most governments offer these vaccines to their people for free or for subsidized costs. After the licensure of the vaccine, you can go into what we call a phase 4. And a phase 4 is what we call post-licensure assessment or surveys. And this is basically because this vaccine is going to millions of people or hundreds of thousands. You still collect data so that you can see how the vaccine is working. You can see if there are any issues with safety or any extremely rare conditions. Because if something occurs in 1 in a million people, for example, then you need very large data sets to analyze that. So that is generally the process of making a vaccine. Now, in all these stages, we have what we call a placebo, which is really something we use to help us to compare the effect of the vaccine and if there was no vaccine. But because we use the placebo to make sure that people getting the vaccine don't know they're getting the vaccine or getting the placebo, it helps us to have an unbiased view, so that we know this effect, if it's a side effect or it's a benefit, is purely because of the vaccine, not because of a random chance. But we do that using placebo, because a placebo could be something like water for injection. It could be an unrelated vaccine that is licensed already. So you give that, but you don't tell the volunteer what they've been given. You just tell them they've either gotten the vaccine or the placebo. So at the end of the day, when you are reporting, you're not biased to say that the vaccine is working, yet it was a random effect. Thank you.
This is TV47. Happy holidays.
The Senate decides to hear impeached Nairobi Governor Mike Sonko in the plenary. Also tonight, cancer patient in Machakos County left seeking treatment at Machakos Level 5 Hospital, sent back home as healthcare workers strike enters day 3. Plus, even religious leaders cannot strike a common accord on BBI. Catholic bishops call for consensus as NCCK calls for a total halt of the process.
TV47. Happy holidays.
So what happens after you actually have a licensed vaccine? Like now we are seeing a few COVID-19 vaccines getting to the level where they may get some form of conditional approval for use, for general use. We've seen the UK has already licensed one vaccine. There are a few others in the pipeline that could get conditional licenses for use in the general public. But this is just the beginning again. So once you have a vaccine, their first challenge is going to be manufacture. The world has close to 8 billion people now, and if everybody could get this, that would be ideal. But we know we are not going to come up with 8 billion doses. Actually, most vaccines require 2 doses, so we are not likely to come with 16 billion doses in a year. And because of that, there will be a challenge in manufacturing. Luckily, because there are several vaccines, maybe 3, 4, or 5 that are likely to get licensed in the next maybe few months, we will have different companies working on this. And we've seen a lot of global solidarity around this, and hopefully we'll be able to make enough doses to give those who require the vaccine the most. And once you overcome the challenge of manufacturing, the second challenge, of course, is access. How do we get these vaccines to the people who need it the most? This is affected by things like finance. Who can pay for it? How is it paid for? So if, for example, we want to take this to the least developed countries who are still having COVID, how do we pay for that? How do we get the poor in these countries who may not afford or who may not be able to pay the vaccine but require it to get it? So access is likely to be a headache for most governments, especially in Africa, including Kenya. And again, there is need to discuss that and see the best financing methods for that. We also have, besides access, challenges of distribution. That yes, even if the country has received enough doses of the vaccine, say we agree to give all healthcare workers and frontline workers and people with underlying conditions and people above, say, 60 years of age, how do you get to these people? We know they are distributed across the country. Most of the vaccines that are in the front line right now are likely to require refrigeration and others freezing, which is very low temperatures, some as low as negative 70 degrees. There are very few hospitals that will have such freezers. So how do you get the infrastructure in place to take these vaccines to where the people who require them are? So that distribution is a challenge, but we know it can be done. So it's just a matter of getting the right resources and the right people to do that. We've seen it happening, for example, with the soft drinks and even hot drinks that are taken to all parts of the country in very good condition. This is slightly different because we're using frozen, a product that requires to be frozen. Let's see if, for example, if that is a vaccine we choose to go by, that would be a challenge. Luckily, we are likely to have other vaccines that maybe in a year or so may not necessarily require to be frozen or may require just the usual refrigeration, and that might make the challenge ease off. Despite having this in place as well, the vaccine may get to the facility where it is required. But there's a third challenge, again, for acceptability and uptake. So do the people who need to have this vaccine actually go ahead and have it? Because we've seen once in a while people refuse to take vaccines because of concerns, many times misinformed, but that cannot be wished away. That if somebody doesn't want to take a vaccine, you really cannot force them. So acceptability of the vaccine, uptake of the vaccine can sometimes be an issue. And this is what we need to educate the public on, that even as the vaccines come in, that by the time the Kenyan government takes up a vaccine, they have looked at the data and they are convinced that the vaccine is safe for use by the general public. The other challenge you might need to think about now is that these vaccines have been tested in adults, because when you're doing testing a new product, you have to do this in people who can give consent, and these people are above 18 years. So it means we may not immediately have vaccines to protect children. Of course, the data for children can be collected much faster once the vaccine is shown to be safe in adults, but means at the outset, when you start to give this vaccine, you cannot give it to children. So we still need to think about how we'll continue to protect our children without necessarily exposing them to vaccines that have not yet been tested in children. So this is likely to delay the vaccines for children. But the good thing is that the data, that once the data that is being collected in adults shows that they are safe, we are likely to have this tested in children much faster. But it is difficult to do that from the start unless you show that the vaccine is safe in adults. We do this in research because you do not want to expose vulnerable people to a risk that you cannot tell. So we start with adults who are more understanding, and they're likely to take up this risk with full consent. And then after that, we may then test the vaccine in children and see if it works. So this is something that may be a challenge, but given the speed at which COVID-19 vaccines have been developed, we may see a fast tracking again of testing in children, of course, without exposing them to undue risk, so that we have a vaccine for everybody. Well, it's true that the efforts to develop an HIV vaccine have taken well over 30 years. And one of the things we need to understand is that although we still do not have a vaccine for HIV today, we've learned a lot from the work that has been done over these 3+ decades. HIV as a virus is very different from the virus that causes COVID-19. HIV, for a start, although all viruses will mutate, they will change in the course of time, but HIV is one of those viruses that is very diverse. It has very different ability to change and to mutate compared with other viruses. So you see HIV as a virus has, first of all, is 2 species. There are HIV-1 and HIV-2. And within HIV-1, which is our biggest problem, we have very many subtypes. And these subtypes require a different immune response. So if you make a vaccine that only covers one type, and as the virus mutates, you will not be able to prevent viruses of other subtypes because they require antibodies that can bind specifically to them. So HIV, because of the ability to change, the ability to mutate over and over, has been a challenge. Of course, the science is trying to catch up with this, and the lessons we've learned over the last 3 decades or so are being employed to make better vaccines, more versatile vaccines that maybe in the near future will help us get an HIV vaccine. This would be good news because we know HIV treatment has worked very well, and if we could get a vaccine, this could be the last kick we need to eradicate HIV from the face of the world. So why is there so much effort in making a vaccine? We know that vaccines are the most effective method of preventing infectious diseases, and we have good examples to show for it. Smallpox, which had been sickening people for thousands of years, was eventually eradicated in the 20th century by use of vaccines. This was because smallpox, at the point that it was eradicated, we were able to focus on areas where the virus was occurring and give people vaccines, and by that eradicate the condition from the world. We've seen polio is actually on the verge of eradication as well. In fact, recently the World Health Organization pronounced that Africa had eliminated the condition in this continent. And this is because polio only currently infects man. So if you give people the vaccine, then it means if everybody is protected, then there is no one who can be infected. And so the virus will literally just die out. And this is the same thing with measles, which 20, 30 years ago was causing a lot of sicknesses, a lot of even death. But with time now, improved vaccine coverage, we are seeing that measles is also gone down considerably. And if we can continue to improve the coverage of measles vaccine, so that we give most people, maybe over 95% who require this vaccine, get it, then we will be able to possibly also eradicate measles. So vaccines work, and this is the reason that we are putting so much effort and emphasis on vaccines, because once you vaccinate somebody and they are protected against a certain condition, then you don't need to think about treating them. You don't need to think about worry about that person infecting others, because other people are also protected. So vaccines are very effective, and for this reason we need to encourage